Hey everyone, welcome to lesson 4 of Bio 205. Today we're talking about prokaryotes, where they live, how they relate to other organisms in their environment, and also a little bit of how we classify these organisms. When it comes to the classification, like a lot of things in biology, it is very intensive in the amount of information there is. So I'm going to be brief on that. There's more detail in the lesson documents, and you'll have to do a lot of review, a lot of contrasting, but it should come over time and over reading those documents of how we specifically classify organisms. And of course, in the real world, you're not going to be tested to identify a microorganism from a batch of tests without any reference. In the real world, there's actually flow charts for testing and how you progress through those different tests when you start them, etc., to get the answer for what organism, at least what organism type you're looking at. This is most common in hospitals, so you'll do a lot of different tests for a bacteria, let's say, that you don't know what exact bacteria it is, but you need to know what intensive antibiotics to start a patient on. You're going to go through essentially a flow chart of different growth medias that you're going to put it in, different tests you're going to do to get to the ultimate answer of what antibiotic do we use. A lot of times that's not even going to tell you the exact microorganism. It's just telling you a classification. And then with that classification, you know what antibiotics work on that entire group. So when it really comes to doing detailed classification, that's when you have genetics come into play and looking at the actual genes involved and using the microbe genome to determine what exact microbe that is. But I want to roll back kind of to lesson 1 and lesson 2 and talk about prokaryotes and the fact that they are, again, everywhere in your environment. So they're going to be absolutely everywhere in your environment, in you, on you, around you, on the walls, in the ceiling, in the floorboards, wherever it is, the ocean, hydrothermal vents, wherever it is, they're going to be there. So they live everywhere, but they live in these different places in different ways. And this is through something called symbiosis. So it's how they live in their environment. And this is really important for human health and animal health and even environmental health, how these microorganisms interact with the other parts of their environment. Usually it's broken into host plus organism. So there's going to be some sort of host that it's interacting with. When we think about a host, it's a higher order organism for the most part. So a human or an animal or a plant that these microorganisms live on. Keep in mind that in that interaction, there's also always going to be this layer of other microorganisms that they're competing with, that they're synergistically living with in that situation. But when we discuss symbiosis, it is usually in that host plus singular microorganism context. Now, there's four main types of symbiosis. One of them is mutualism. This is pretty much what happens with the gut microbiota with humans. You have a microorganism or an entire group of microorganisms that live in your gut. They get to live in a place where, you're eating, you're drinking normally. They get food and nourishment from that process. And in response, we get a protective layer. We get chemicals that actually, go up to our brain and make our brains happy through just having this diverse microbiota in this mutual beneficial situation where they're living off us, but we're also getting something back. So that's the mutualism. It's a sort of quid pro quo where we give them somewhere to live and they give us a benefit from living there. There's also commensalism. So commensalism is where the bacteria, using the gut microbiota example, lives in us, but they're actually not giving us any benefit. So it's where the microorganism is in the host, on the host, and doesn't do anything detrimental, but is kind of just taking up space, like a mooch essentially of the microorganism world. So they're living there, they're getting all the benefit that those mutualism organisms are getting, but because they're commensal, they're kind of just hanging out, but they never harm us. So it's okay. They're there, they're just in that space that might be taken over by a more beneficial organism, but it could also be taken over by a organism that's going to harm us. And this gets into parasitism. So parasitism, like the name suggests from parasites, is a microorganism that is living in a host and taking away from the host. So if you've ever had the stomach flu or an infection that require antibiotics, those are parasitic microorganisms and viruses that are inside you, and they are taking away your resources without giving you anything back. And that's why it leads to sickness and is really, really bad, and why we want to clear those out and get rid of them and be healthy again. There is also a last group called amensalism, and that's when the host is getting a benefit to the detriment of the microorganism. So this is a little bit newer on the front end of microbiology because it was always thought that there was mutualism, commensalism, parasitism because it was very host focused. But there is this group where you're going to get a reaction from the host that makes it so you're getting a benefit from the microorganism to the detriment of the microorganism. And usually this is in the context of killing. So if you have, microorganisms on your skin, you have plenty of different defensive layers on your skin that will attack that microorganism and clear it out and get rid of it. But in some cases, you're also taking with that some sort of benefit from the microorganism. This is less in a human context and more in a plant context when that happens, but it does happen for sure. Now, with all that in mind, with that symbiosis structure in mind, it's also important to understand how microorganisms react to each other in the food chain. So, one of the main differentiators is there's going to be producers and consumers. So, autotrophs and heterotrophs. Autotrophs are the producers. They're going to make essentially energy from something like sunlight or heat or radiation. Extremophiles exist that do make different growth factors. They're growing, they're fixing carbon, they're reproducing based on not just sunlight, but those extremophile situations where it's heat, it's chemicals, it's electricity, it's radiation. So you have these groups that are essentially taking something in that's innately in the environment and unprocessable by most microorganisms, like sun, and producing themselves. So they're replicating, they're growing, they're reproducing and creating what we call biomass. And then there are the heterotrophs or the consumers that come along and are the predators that eat the biomass to themselves grow. So in the case of humans, we are heterotrophs. So we consume non-innate energy to grow. So we have to consume life to live, whereas a algae, for instance, that's getting most of its energy from the sun and different chemicals in the ocean, they're able to live in this autotrophic world where they're just producers. They're just making more of themselves. They're just growing, and then other things come along and eat them. So that's really important to understand. And this feeds into all that symbiosis because there's bacteria that are eating other bacteria. There's bacteria that are in our guts eating things that we can't eat, we can't process. So they process it either just completely and keep it to themselves, or they process it in a helpful way and they give us part of that digested, previously unprocessable nutrient back to us. So there's all of these things that are happening in that food chain. And there's also, of course, competition. There's some, autotrophs that have good defenses against heterotrophs that try and eat them. And then there's all this evolution, this arms race for the heterotrophs to come in and eat the autotrophs and get all of their yummy, yummy nutrients. So all of that's going on in this food chain, which influences everything that's going on in those symbiotic relationships, whether it's parasitic, whether it's commensal, whether it's anything else. That is going to influence those relationships just based on what kind of overall producer or consumer the microorganism is. And this gets into habitats. So in the water, there's a lot of free-flowing nutrients. There's, of course, an abundance of water that's there, and also a lot of sunlight availability in the water, especially in the ocean. So you get a lot of autotrophs, which then leads to a lot of heterotrophs. Basically, all sea life follows. You have plankton, which are small microorganisms, and they're eaten by a bunch of different things, whether it's a microorganism all the way up to whales. There's whales that all they do is they filter plankton and eat that as their main source of nutrients because it's so abundant in the ocean. So that's why there's a lot of diversity of life there because there's a lot of availability of these basic building blocks for autotrophs, and then also a lot of autotrophs because of that for the heterotrophs. Soil is similar, so you have, your decomposers that are recycling dead matter, whether it's plants or animals or, worms or other microorganisms. And then you get a nitrogen cycle of nitrogen fixers that take the free nitrogen from the decomposers and put it back into a, concentrated space. So there's a lot of this going on in soil environment where you get the rise, the fall, the decomposing, and the reconstitution. And that's, it's readily visible in soil because you can walk through a forest and fall and you smell that decomposition, that kind of sweet smell because all the leaves are decomposing and that's all the microorganisms churning through those, turning them into dirt and taking all the nutrients from them and fixing them in themselves and then getting eaten by other microorganisms. Whereas in the ocean, that's a harder process to see, but that process is still happening, right? And those are the two main places that we think about microorganisms. They're in the soil, they're in the dirt, they're in the dust, or they're in the water. But of course, they're also in plants, and you have a lot of mutualism going on in plants. In fact, plants can't survive without certain bacteria and microorganisms fixing nitrogen around their roots so that they can readily have nitrogen availability. And there's also a lot of plant pathogens that then feed off of the plant in a parasitic way. Agrobacterium, I worked with a lot. I mentioned briefly in some of the other lessons. If you ever are walking around and you see these big bulbous tumors on trees, that's agrobacterium infecting the tree, forcing it to produce sugar so it can eat and replicate. So you get these very interesting situations where in plants especially you have a lot of parasitic organisms that the plants can't really get rid of, so they just stick with the tree or the plant for the rest of their life until ultimately they die. Of course, you have animals and humans. Those are another main place we study microorganisms in an environment, being us or the animals. And usually this is to study disease, of course, because, we want to be healthy. So we're going to study the bacteria, the microorganisms, the viruses that make us sick. So when we think about animals, there's things like, you know, the black pig, Yersinia pestis, that's born on fleas, gets onto rats and any other animal, and of course then infects humans and causes the black plague. There's a ton of tick-borne diseases, which are important because if you have a dog and they're out and they're running around in your yard and they bring back ticks, not as much of a problem in Arizona because a lot of the ticks can't survive here. But if you're out east somewhere, there's a lot of tick-borne diseases that the dogs will get the ticks. They'll bring the ticks inside. You get bit by the tick, and suddenly, you might have Lyme disease. So we have to think about all these things. And that's actually in the ticks mediated by, of course, a microorganism. It's not the tick causing the disease, it's a microorganism that the tick transfers to us. The same thing with malaria from mosquitoes, for instance. So we think about those things. In humans, there's a lot of talk, especially now, around your microbiota, how you keep that healthy. I actually authored a review paper with a couple colleagues about how important the microbiome is in the ICU hospital setting. So you have people who are very, very sick. You want to make them better. A lot of what they need is really intensive antibiotics, which completely clears out your microbiota and can lead to other issues like C. diff, which is a horrible intestinal infection of a bacteria that is normally commensal. So it lives in you at all times, but if it doesn't have anything else in competition with it, it becomes parasitic. And that is one of the things that is a big worry about, people go in, say, "Hey, I'm sick, I need antibiotics." And okay, you get the antibiotics, but if it doesn't clear things out, then you need better antibiotics. And if you keep getting sicker and sicker, you land in the hospital, you get cleared out with your antibiotics, you're better from the initial disease, but then you get something like C. diff. How do we prevent that? How do we bolster the microbiome that you have to prevent that secondary hospital infection, right? So all of this is really interesting and it gets studied pretty in depth nowadays. And that study is facilitated mostly not by the classical classification methods that you're going to learn about in this lesson, but by things like genomic uh, mass studies of intestine samples. So you take someone's sample, you do a large screen, you find a bunch of different diverse microorganisms in your microbiota, and then we try and learn from that person's health profile and a couple hundred other people's health profiles who has good microbiome, bad microbiome, which bacteria are good, which are bad, which are commensal, which are mutual, which are parasitic. And we're starting to really figure all that stuff out now, and it's really interesting, but it kind of has to be done through that massive genomic study because growing up the microbes and seeing that diversity is very, very difficult. But it's also very important to have all of these different ways of classifying microbes and also different tests we can do that give us rapid turnaround of which microbes we're dealing with, especially again in a hospital setting. So someone comes in, you don't know what they have. They've done a round of antibiotics that typically works on, let's say, some sort of stomach infection. Then you have to take samples, do a bunch of lab tests to look at, okay, what are we going to actually be dealing with here? What antibiotics work really well on this? How do we not blow out that person's microbiome with antibiotics? So we want to be targeted. We don't want to be as broad as possible. And that, of course, gets into personalized medicine and doing what's best for that exact patient versus what's generally going to be done for the vast majority of patients. And that's a little bit outside the scope of what we're discussing. So I want to bring it back and talk a little bit about how we actually do those classifications in a research and a hospital setting. So as I mentioned, the first thing is your genotyping to phenotyping. So you're going to start with a genome that's going to give you a phenotype, and that is what we are studying a lot of the times when we're doing our genome analysis. So we're actually taking all the DNA, we're looking at it all, we're saying, okay, this is this type of microorganism because of these different genes it has. One of the issues with this, though, is it's very hard to discern what level of difference between different microorganisms means it's a new organism, right? Because organisms are defined taxonomically. We talked about the taxonomic classifications and how that comes about mostly through looking at what that organism has. Does it have a flagella? Yes, no. Is it growing in this type of media? Yes, no. Does it have cell walls? Yes, no. So we're figuring all of that out, and then we have all this genomic information that maps on top of it. But, every microorganism has a slightly different genome. That's how evolution works. There's mutations, slight differentiation, and then at some point, that slight differentiation wins out in the competition of life, and we get new microorganisms over a period of time due to that differentiation. But if you take a section at any point, you're going to see some slight differences, even though it's technically the same microorganism. It hasn't undergone enough changes to be new. So we have all this genomic information, but there's also the importance of that phenotype. And the phenotype is what's studied through all of these classical classification methods. And that's your microscopy and stain. So you learned about that in last lesson. The type of microscopy you use is very important. The type of staining you use is very important. And figuring out what a microorganism is based on that visual classification is always going to be needed because genetics can only take you so far. It tells you a lot, but at the end of the day, you need to confirm with some sort of microscopy or some sort of different test. There's also, you have different culture traits. Now, this is culture not as in like cultural, but culture as in a growth media. So a growth media is called a culture because you're growing bacteria in it. I honestly don't know why it's called a culture, but that's the term that's used. That's the scientific term. So you're growing organisms in different medias, different cultures, and that is giving you a window into what environments can they live in, where do they survive. So, going back to agrobacterium cannot grow at human temperature. So I'm sure that someone at some point has like eaten some agrobacterium on accident, whether it's from, like cutting open a tree or in the lab and accidentally, not wearing the right PPE and contaminating themselves. But it can't grow at human internal temperature. Therefore, if you're growing it in the same culture as E. coli, you can check if it's a contaminated culture with E. coli by just sticking it in at human temperature in an incubator. And if it all dies, it's not contaminated. If something else grows, it's contaminated. Could be E. coli. E. coli's everywhere. So it is probably something like that that's growing there. So it's very easy tests like that that allow us to differentiate different organisms in a way that doesn't take all this genomic work to do. Same with microscopy, of course. And then there's a lot of other tests. There's a lot of fermentation tests, enzyme assays, and I also, growth requirements goes beyond the temperature. It goes into what exactly you're putting into the growth media, that cultural media. Is it going to be in need of CO2? Maybe. Is it going to be in need of a very, very nutritious base, something like fetal bovine serum, which is just blood from a young calf? Is it going to be in need of only yeast extract? Yeast extract is used a lot because that's a very diverse nutrient base. Or can it get away with just some very extreme things being thrown in there? You put some different salts, you put some different base building blocks of nutrients, and then you expose it to light. Does it grow in that? Is it photosynthetic? Is the question you'd be asking there. Yes, no. So a lot of that is done in these culturing experiments. And there's also motility experiments. If you put your bacterial sample in agar, it stops it from being motile just by, shaking around and stuff. But motile bacteria can swim out of that agar and then get up to the top where all the nutrients are and then spread into the actual nutrient section. So you'll usually have like an agar base, and then up here you'll have a liquid, and you do the agar, you pour the liquid on top, and if it spreads up into that liquid, it's a motile bacteria or at least a motile microorganism. So there's all of these interesting tests that we can use. Other things are reproduction. We can study that as well. So if you're looking at, E. coli can double in 20 minutes, so it can grow overnight. You can put a sample with inoculated, and that means inoculated means you're adding E. coli to a culture. So you inoculate a culture with what you think is E. coli, it should grow overnight. It doesn't, but it grows in 2 days. Okay, what happened there? Is it actually E. coli? Let's do some other tests and figure that out. So again, all of these tests are very important, and you're going to use these tests to define specific microorganisms. That is so expansive that it's really hard to cover outside of a textbook. And in some labs, they literally have flowcharts on the wall of you do this, yes, no, if it does grow, then you need to do these follow-up experiments. If it doesn't grow, then you need to go back and you need to grow in these media to check. And all of that sort of stuff is pretty cool, but at the same time is very hard to explain just talking. It really is something that you have to sit down and review, and that's something that is going to be very important for this lesson, is to sit down and review all these different culture types, all of the different microorganisms that can be grown in certain cultures, and sort of setting up that mental flow chart of, okay, I know again, E. coli grows at 30 degrees Celsius. And Agrobacterium will not grow at that. It'll die. So I have two cultures. I think they're contaminated with E. coli. They're supposed to be Agrobacterium. I'm going to put both of them into an incubator at 30°C. One of them grows, one of them doesn't. The one that doesn't, I know should be agro. The one that does grow, I know is contaminated with something. It's probably E. coli, I would say, but it might be something else. So that is a very easy experiment to do. And you'll kind of get a feel for that as you're going through the lesson. And then another thing is when it comes to extremophiles, those are actually very difficult because archaea, remember we talked about them in the first lesson, they are extremophiles at baseline. That's one of the things that we use to define them. They like to be extremophiles. So something like extremely acidic environments, you might not get bacteria to grow in, but you can get archaea to grow in. Uh, if it's high salinity, so the Dead Sea, for example, is like 35-ish% salt in that water. That's extremely salty water. And there's extremophiles that live in there, and those are generally going to be archaea. So you can do other experiments where you're growing in these extremophile ways to test if there's archaea there. And that's another subset of doing all of this microorganism testing. But you also have to be aware of the buckets have some overlap, so you can have some other things like bacteria that will also grow under those conditions. And then you have to do further tests to determine if is this really archaea? Is this bacteria? The nice thing about genomics is it's very easy to tell if something's archaea or bacteria with genomics. So that's typically what's done there. So you don't have to worry about anything like that. Now, I hope that's a good introduction to why we do these tests, some of the tests that are done, and kind of building this mental flow chart of the different tests you'll do under different scenarios. It's a lot of context for what will be a lot of information. So the information can seem very overwhelming, but if you just go through it step by step, do some review, and think about it from a research clinical point of view of, okay, I have these tests that help me classify these things. I have these other tests that can be run to help classify these things. It breaks it up in a sensible way that I think is very intuitive over time. Now, uh, what you're going to want to be able to do at the end of this lesson really is explain what symbiosis is, the different habitats of microorganisms, which is what we really touched on in this video lecture. But then you're also going to be want to be able to, excuse me, to outline these classification logical steps. And that's where there's going to be a lot of work on your part because it is all written as I mentioned, and that's really the best way in my opinion to go through it. But as always, if you have any questions, please just let me know, and I am always a resource to help you get through anything that is maybe a little bit overwhelming, a little bit nonsensical. Science can be like that a lot of the time in the naming conventions and the way things are described. So if you're needing any help anywhere, please feel free to message me. And I wish you the best of luck diving into this lesson. Have a good day.